Plastids are a diverse group of membrane-bound organelles found in the cells of plants, algae, and some other eukaryotic organisms. They are essential for various metabolic processes, including photosynthesis, pigment synthesis, and storage of vital compounds. All plastids share a fundamental double membrane structure known as the plastid envelope, which encloses their internal components. This envelope consists of outer membrane, a semi-porous membrane that is permeable to small molecules and ions but less permeable to larger proteins. The inner membrane is, well, a more selective membrane that regulates the passage of molecules into and out of the plastid. Intermembrane space. The thin space, about 10 to 20 nanometers, located between the outer and inner membranes. Within the inner membrane, the aqueous fluid matrix is called the stroma. The stroma contains plastid DNA, which are circular double-stranded DNA molecules similar to prokaryotic DNA, reflecting their evolutionary origin from endosymbiotic cyanobacteria. Ribosomes, specifically 70S ribosomes, are smaller than eukaryotic ribosomes and also similar to prokaryotic ribosomes. There are various enzymes involved in metabolic pathways, such as the Calvin cycle in chloroplasts. And lastly, storage granules, which, depending on the plastid type, can include starch granules, lipid droplets, or protein crystals. In some plastid types, particularly chloroplasts, there is an additional internal membrane system called the thylakoid membrane. This membrane forms flattened, sac-like structures called thylakoids, which are often stacked into structures called grana, with the singular being granum. The space inside a thylakoid is called the thylakoid lumen. Plastids are a diverse group of membrane-bound organelles found in plant cells and some other eukaryotic organisms like algae. They are essential for various metabolic processes, including photosynthesis, food storage, and the synthesis of important biomolecules. All plastids share a fundamental double membrane envelope consisting of an outer membrane and an inner membrane, separated by an intermembrane space. Inside the inner membrane is a semi-fluid matrix called the stroma. While the basic envelope structure is common, the internal membrane systems vary greatly depending on the type of plastid. Outer membrane, permeable to small molecules and ions. Inner membrane, more selective, regulating the transport of substances into and out of the plastid. The intermembrane space is the region between the outer and inner membranes. The stroma is the aqueous fluid filling the plastid, containing enzymes, ribosomes similar to those in bacteria, circular DNA known as plastid DNA or cpDNA, RNA, and sometimes starch granules or lipid droplets. This DNA, by the way, reflects their evolutionary origin from endosymbiotic bacteria. 3. In specialized plastids like chloroplasts, additional internal membrane systems are present. Thylakoids are flattened, sac-like membranous structures. In chloroplasts, these are the sites of the light-dependent reactions of photosynthesis. Grana, which is the singular form of granum, are stacks of thylakoids. Stroma lamellae, also known as intergranal thylakoids, are unstacked thylakoids that connect different grana. Plastids are complex organelles composed of various biochemical molecules. Proteins make up a significant portion of the plastid's dry weight. These include enzymes for various metabolic pathways, such as Calvin cycle enzymes in chloroplasts, structural proteins for membranes, and ribosomal proteins. Lipids form the membranes, which include the outer, inner, and thylakoid membranes. These lipids include phospholipids, galactolipids like monogalactosildiacylglycerol and digalactosildiacylglycerol, and sulfolipids. Pigments are crucial for light absorption in photosynthetic plastids. Examples include chlorophylls, such as chlorophyll A and B, which give green color and are vital for photosynthesis. Carotenoids, including carotenes and xanthophylls, impart yellow, orange, and red colors and also play roles in photoprotection. Plastid DNA, or cpDNA, is a circular double-stranded DNA molecule that carries genes for some plastid proteins, rRNAs and tRNAs. RNA within the plastid includes messenger RNA, ribosomal RNA, and transfer RNA, all involved in protein synthesis. Starch granules can be found in the stroma, serving as temporary or long-term energy storage. Ions and water are essential for maintaining the osmotic balance and facilitating enzyme. Plastids are broadly classified based on the presence or absence of pigments and their primary functions. All plastid types are believed to originate from proplastids, undifferentiated plastids found in meristematic cells, and can often interconvert depending on cellular needs and environmental cues. Proplastids structure small, undifferentiated, colorless plastids with a simple internal structure, location found in meristematic tissues which are actively dividing cells of plants. Function, precursors to all other plastid types. They are responsible for the continuity of plastids across generations. Chloroplasts, structure green, typically discoid or oval-shaped organelles with an extensive internal membrane system of thylakoids stacked into grana. Location found in photosynthetic tissues, primarily in the mesophyll cells of leaves. Composition, rich in chlorophylls, carotenoids, enzymes for photosynthesis, plastid DNA, and ribosomes. Function, the primary site of photosynthesis where light energy is converted into chemical energy such as sugars. They also play roles in the synthesis of amino acids, fatty acids, and some lipids. Chromoplasts. Structure, variably shaped, often containing irregular membrane vesicles, lipid droplets, or carotenoid crystals. They lack chlorophyll, location found in fruits, flowers, and aging leaves. Composition, abundant in carotenoid pigments like carotenes and xanthophylls, which give yellow, orange, and red colors. Function, provide vibrant colors to attract pollinators and seed dispersers. They're involved in pigment synthesis and storage.
Leucoplasts are colorless plastids, generally lacking extensive internal membrane systems. They are found in non-photosynthetic tissues such as roots, tubers, and seeds. These plastids are devoid of pigments and are primarily involved in the storage of various substances. They are further specialized into different types: amyloplasts, which store starch, for example, in potato tubers and rice grains; oleoplasts or oleoplasts, which store fats and oils, like in oily seeds; and proteinoplasts or aleuroplasts, which store proteins as seen in some seeds. Idioplasts are pre-chloroplasts found in plants grown in the dark. They contain a semi-crystalline prolamellar body, which is a lattice of tubular membranes instead of organized thylakoids. These are located in the shoots of seedlings grown in darkness. Upon exposure to light, idioplasts rapidly develop into chloroplasts, as the prolamellar body reorganizes into thylakoids and chlorophyll synthesis begins. Gerontoplasts are degenerating chloroplasts. They show a breakdown of thylakoid membranes and chlorophyll. These plastids are found in senescing or aging leaves. Their function is involved in the breakdown of photosynthetic machinery and the recycling of nutrients during leaf senescence.